So the function of the TRPR gene is to make mRNA then that codes for making the repressor. This repressor, though, look at here, is made in the inactive form. So what does that mean when the repressor is made in the inactive form? The word repressor means that it has the ability to repress the genes or turn off these genes. It has the ability to do that. And so if the repressor is in the inactive form, what does that mean in terms of what can't it do? It can't repress. Why can't it repress the genes or turn them? It has to do with the shape. So the shape is not the right shape. And what's the right shape? Like, why is this the wrong shape? What can it do? It's in the wrong shape to do what? Bind to the operator. And so therefore, when it can't bind to the operator, then RNA polymerase is able to bind, and therefore these genes are able to be transcribed and translated, and therefore you make the enzymes to make tryptophan. So these genes, since the repressor is made in the inactive state, normally these genes are on or off. They're on because the repressor is made in such a state that it can't bind and turn the genes on. So these genes are normally operating on. All right. So that's the regulatory gene. So then I ask you what structural genes are included and what do they make? So the structural genes are the genes that are part of the operon that are being turned on or off. So in this case here, there's 5 genes. They just generically named them TRPA through E there. And each one of these genes corresponds to make a different enzyme. So we have enzyme A, enzyme B, C, D, and E. So we're making these 5 enzymes. This is beneficial for the bacteria because in order to make tryptophan, they need all 5 of these enzymes. So therefore, it makes sense that we want this, when this is turned on, we want all these genes to be turned on together, so we make all 5 enzymes so then we can make tryptophan. It would not be beneficial for the bacteria just to make enzyme A, because enzyme A wouldn't be enough enzymes to make tryptophan, and so therefore you need all of them on. So therefore, you have one promoter region that turns all of these 5 genes on at once. And so your genes are TRPA through E, and your products are enzymes A through E. All right, the next question asks you, is the operon inducible or repressible? Let me review what those words mean. All right, so inducible has the ability to be induced. In this case here, it has the ability to be turned on. So these genes have the ability to be turned on. When we say that they're inducible or have the ability to be turned on, what does that mean that they usually are then? Off, right? And then if it's repressible, means it has the ability to be repressed or the ability to turn off the genes, which means the genes are normally on, right? So therefore, the Trp operon, these genes are they normally on or off, these genes? They're normally on. The activator is made in the inactive form. It can't bind here. And so these genes are normally on. The RNA polymerase can go and bind and make these enzymes to make tryptophan. So they're normally on, so therefore they have the ability to be repressed, which means that it's a repressible. All right? All right, so then is the repressor protein produced in the active or inactive? So we've gone over that. That's the inactive form. And then the repressor protein becomes active when it interacts with, so this repressor can become active when tryptophan is present. That's what this picture here shows you. If tryptophan is present, tryptophan binds, puts the repressor in the active form. The repressor then can come and bind to the operator. RNA polymerase can't bind, and therefore it turns them off. And so, and that makes sense because the bacteria, if the bacteria is given tryptophan, if tryptophan is in its environment, then it would be very wasteful to make tryptophan when it's given to you. So therefore, if tryptophan is present, the genes are turned off, it doesn't make tryptophan until there's no more tryptophan in their environment. All right, so that's the right-hand side with the Trp operon. So that was anabolic because we're making tryptophan. On the flip side, the Lac Operon, the Lac Operon catabolic, we're going to break down lactose. Let me review what lactose is. Lactose, remember, is a disaccharide. It's a glucose hooked to a galactose. They hook together, so it's a disaccharide. So bacteria can use lactose for energy. They can take it into their cell and use it, but what they have to do to it is they can't use it in the lactose form. What they have to do is break it down into glucose and galactose. So they have to digest it. All right, in order to do that, to break that bond, they need enzymes. So therefore, they only make those enzymes if they have lactose present, because it would be very wasteful to make the enzymes to digest lactose if there's no lactose in their environment. So that's the background there. So when I asked you for the regulatory genes here, this is the Lac I gene. It makes mRNA to make your repressor. This is a little bit different. The repressor in the Lac Operon is made in the active form. So if it's made in the active form, what does that tell you about whether or not these genes are normally turned on or off? They're usually off. So like over here, since the regulatory gene makes the repressor in the active form, the repressor can bind to the operator, which means RNA polymerase can't transcribe the gene, so these genes are normally off. And so, so what genes are we talking about? So the next row asks you about the structural genes. So this is the generic names Lac Z, Lac Y, and Lac A genes. Lac Z makes, they each make a different enzyme involved in the metabolism or breaking down of lactose. So beta-galactosidase, permease, and transacylase. So those are your structural. Then I asked you, is the operon inducible or repressible? So since the operon, the repressor is made in the active form, so these genes, these genes are normally off. That means that then this is an inducible, they're able to be turned on or induced. And so the repressor for the next question is made in the active form. That's why it's inducible. And then the last one asks you, the repressor protein becomes active when it interacts with, kind of a trick question here because it's nothing. It doesn't need to interact with anything because the repressor is made in the active form. So it's made in the active form, which means that the genes are normally turned off. It doesn't have to interact with anything to become active. It can interact with allolactose, which is an isomer of lactose, but that doesn't convert it to an active form. It converts it to inactive. And that makes sense. Why? Because allolactose, you only have allolactose if lactose is present. So we only want these genes to be turned on if lactose is present. And so if it is, binds to the repressor, makes it inactive, and that repressor can't bind to the operator, and so therefore these genes are turned on, and so these genes are turned on only when lactose is present. All right, and so then the bacteria is not making enzymes to digest lactose when there's not any present. All right, so that's nothing there. New questions on it? Okay, so what we're going to do, so that was a quick little review of what we did on Wednesday. We're going to finish the last little bit of 18.4, which is why I wanted to leave with the end up with the Lac Operon, because the last little bit of 18.4 adds another layer onto the Lac Operon. So the Lac Operon has one more thing that helps to control whether or not this is turned on or off. So get out your 18.4 notes if you don't have them. So the Lac Operon is only turned on when lactose is present, which remember lactose, right on the board here, lactose is a disaccharide, so it is glucose hooked to a galactose. And here's your little bacteria. So what we've learned is that the bacteria will only turn these genes on if lactose is present, so it's not wasteful. The thing is, if the bacteria has choices, so lactose, if it's digested, we can break it down into glucose and galactose. What do we use that for? Or what does the bacteria use that for? For energy, like cellular respiration and so on. Okay, so it uses it for energy. And if the bacteria has glucose as well, so now in its environment, it has, this is what I mean by choices. It has glucose available and lactose available in its environment that it can take in and use for energy. Which one would be more efficient for the bacteria to use, do you think? Glucose or lactose? What do you think? Alan? Glucose, why? That's correct. In order for this bacteria to use lactose, it can use lactose, but it can't use it in its current form. It would have to make the enzymes to break the bond between the glucose and the galactose, and then it could use those monosaccharides for energy, but it can only do that if it makes those enzymes. Versus glucose, glucose can be taken right into the bacteria and you don't have to do anything to it. It's already in a monosaccharide form. It can be broken down to make ATP for energy, and you don't have to do anything to it. So, therefore, if given the choice, even if lactose is available to the bacteria, it will choose glucose. Now, what we're going to look at is how it does that. It doesn't think, ooh, there's glucose and there's lactose. Let's take glucose because that's going to be more energy efficient. So how does the bacteria know if there's glucose present or not, and use that over lactose? But I forgot, we didn't do the last. We have a couple bullets before we get to that, I think. I forgot. You have 3 bullets before you get to this section called positive gene regulation. The first 2 bullets, see if you can fill those in based upon what we just did in that chart. All right, so the first one, inducible enzymes usually function in its catabolic pathways. So catabolic means we're breaking something down. And so inducible enzymes means that enzymes are able to be turned on. They usually function in catabolic pathways. And so in this case here, would the Lac or the Repressor Protein be an example of that? Do you think? The Lac or the trip? Reducible enzymes functioning in catabolic pathways. Catabolic pathways, we're breaking something down. Reducible enzymes means that the genes are able to be turned on to make the enzymes to break something down. This is the Lac. All right, so this is example Lac Operon. And then repressible enzymes usually function in anabolic pathways. So in this case here, that would be the Repressor Protein. All right, so this other layer we're going to look at with the Lac Operon. So what we've talked about so far, regulation of the Trip and Lac Operons involve what we call negative control of genes. Why is it negative? Because the operons are switched off by the active form of a repressor. So a repressor has the ability to repress or turn off the genes, but the repressor has to be in that active form. So it's negative because we're talking about this molecule turning off the gene. We're going to learn now that the Lac Operon, you not only for the Lac Operon to be turned off or on, you have to have an inactive repressor, so it can't bind to the operator, but you also have to have what's called an activator. So a repressor, when a repressor is active, it represses or turns off the gene. We're going to learn about an activator. So some operons need an activator to turn on a gene. So this is called positive gene regulation. Some operons are also subject to positive control through a stimulatory activator protein such as metabolite activator protein. We're going to call it CAP. An activator protein has the ability to turn on a gene. Okay, so that's what the activator means, is the ability to turn on a gene. When I say ability, that means it has to be in the right shape to turn on the gene. Okay, so I'm going to write that here, ability to turn a gene. Or in this case, operon on. Bless you. So, as I was talking about with the bacteria, with the choices of lactose and glucose, when glucose, a preferred source of E. coli is a type of bacteria, is scarce, the Lac Operon is activated by the binding of this Cap protein. When glucose levels increase, the cap detaches from the Lac Operon, turning it off. So basically, the bacteria needs to turn on the genes to digest lactose when two things are true. When lactose is present, so otherwise it'd be wasteful to make the enzymes to digest lactose if there was no lactose. So lactose is present, but also when glucose is absent, because if glucose is present, it's not going to digest the lactose, it's going to use glucose because that's more energy efficient. So therefore, those two things have to be true to turn the Lac Operon on. Lactose is present, glucose is scarce, or there's not a high quantity of it. So let's look at this picture here that shows you the Lac Operon. So this is kind of like what we've talked about before. So here's the regulatory gene, the Lac-I gene that makes the repressor. Here's your promoter region with your operator, and then this is the first of your Lac genes that are going to be turned on or off. Okay, so let's look at the situation now. The bacteria, this is true about the bacteria's environment. So lactose is present, so it has lactose, and glucose is scarce, so it doesn't have glucose. So it has lactose available to it, doesn't have glucose available to it. Do we want to turn the Lac Operon on in this case? Yes. You want to digest lactose to get energy for the bacteria because glucose isn't an option in this case. So how does the bacteria know when glucose isn't present? Let's make some enzymes to digest lactose for energy. So this is how it knows. So notice here glucose is scarce, and in parentheses it says cyclic AMP or CAMP. The levels are high. So let's talk about what that is, cyclic AMP. We've talked a little bit about it before. Glucose. Glucose is used to make ATP, right? Remember, we break down glucose to put the phosphate back onto ATP. So let's look at, if we use ATP, it breaks down into ADP and inorganic phosphate, right? And that's an exergonic reaction that releases energy for cellular work. Normally, if glucose is present, let me get a different color here. Normally, if glucose is present, this reaction can occur in the opposite way. When we put the phosphate back onto ADP to make ATP, that's cellular respiration right there. So going in the opposite direction, putting the phosphates back on. So cellular respiration regenerates and makes ATP. But in order for cellular respiration to happen, what do you need? Glucose, right? Do we have glucose? No, glucose is scarce. So therefore, what's going to happen, I'm going to make this arrow here to the right bigger, because what's happening is ATP is being broken down into ADP and a phosphate for energy, but since glucose isn't present, the reverse isn't happening, so they're not regenerating ATP. So when a cell is in dire straits like that, they can take the ADP and further break it down for energy. If we further break down ADP, it can break down into a MP, right? And so in this case here, ADP can break down into AMP if there's no glucose present. And AMP can be converted to cyclic AMP, so whenever you have no glucose, this is what's going to happen, and you're going to get your levels of cyclic AMP go up. So no glucose, a lot of cyclic AMP. So what does cyclic AMP do? So if there's no glucose, we want to turn these genes on to digest lactose instead. So here's our cyclic AMP. Here's your inactive cap protein. So the cap, remember, is an activator. What does that mean? It can activate or turn on genes. But it's in the inactive form normally in the cell. Inactive form means that it doesn't have the right shape. So if you look here, cyclic AMP has a shape that's complementary to a shape on the cap protein. So what happens is that cyclic AMP, which is only found when there's what? Glucose is scarce. So when there's little glucose, that's the only time we see that. It binds to the cap protein, and what does it do according to this picture? What does it do to the cap protein? It activates it by doing what? Binding to it, which does what to it? Changes its shape. All right, so the active cap, it's active Y. Whoops, it's supposed to be parentheses. Cyclic AMP binds and changes its shape. So now that's why it's active. So once it's in the active shape, what does it do according to this picture? It binds to the promoter. So there's a special binding site, so it binds here. So this active now can bind to the promoter. It couldn't bind to this unless it's in the right shape. And then when it binds to the promoter, then RNA polymerase can bind and can therefore transcribe the genes. So this is only if cap binds to the promoter. So, cap is an activator. It turns the genes on and allows RNA polymerase to bind. Cap can only bind if it's activated. It can only be activated if cyclic AMP binds to it. And cyclic AMP is only present when glucose is scarce. All right? And so therefore, that's one aspect of turning this Lac Operon on. Because remember, we don't have glucose, but we do have lactose. The second thing is the lactose, and this is the pulling in what we've already talked about. Lactose, remember, some of it gets converted into its isomer called allolactose, which binds to the repressor and makes the repressor inactive. The inactive repressor then can't do what? Can't bind to what? The operator, which means that the RNA polymerase can go and transcribe the gene. So therefore, there's two layers of turning the Lac Operon on. The first is glucose is scarce, all right, and having the appropriate activator. And then the second thing is having an inactive repressor. So you have to have lactose present to make this inactive. And only when those two things, when you don't have a repressor and you do have an activator bound, then those genes are turned on. So let's kind of put that all together here. So this turns on the Lac Operon when, and let's just review here. Let's see if you can come up with two things. So what's the first thing that needs to be true to turn the Lac Operon on? Okay, yeah, lactose present. So let's write lactose present. So if this was like, you had to explain this in an essay or something like that, that wouldn't be enough. The lactose present. All right, yeah, lactose does need to be present. So let's elaborate on this. What does the presence of lactose do? So what does the presence of lactose have to do with turning the Lac Operon on? right? It inactivates the repressor. And so the lactose present, what happens, actually let's put a little arrow here. Lactose present, some of that lactose gets converted to allolactose. So I'm going to put here some lac converts to allo, I'm just going to abbreviate allolactose, which, as Matt says, an allolactose binds to repressor and inactivates it. How does it inactivate it? It changes its shape, right? So why is having an inactive repressor important? What can it do now? The repressor in the inactive form can't bind to the operator, and therefore RNA polymerase can transcribe the genes. All right, so repressor, I'm just going to abbreviate, can't bind to operator, and therefore the genes can be turned on. So RNA polymerase can. So yes, let's say, so RNA polymerase can transcribe the genes. So that's the first thing. So you have to have this inactive repressor so it can't bind to the operator. What's the second thing? There's one other thing that to turn these genes on that needs to happen.
Must not have enough glucose.
That's right. So it must not have enough glucose. So this glucose, either there's no glucose or very, very little, where there's way more lactose than glucose. So I'll just stick with the same wording as your book. It says glucose is scarce. So there's little glucose. So, because it only wants to use lactose and turn these genes on when the glucose isn't present, because glucose is more efficient to use. So glucose is scarce. With having no glucose, what does this do? How does this help turn the gene on? Cyclic AMP. So when glucose is scarce, what happens? Cyclic AMP, what happens to it? There's a lot of it. So glucose is scarce, which means we have an increase in cyclic AMP levels, which, because of what I was just talking about here with the breaking down of ADP. So therefore, these high cyclic AMP levels cause what to happen? What's the CMP can't do? Yeah, so cyclic AMP then binds to your activator protein called CAP. And when it binds to it, it does what? Changes its shape and activates it. So CAP activates by changing the shape. Okay, so now you have this active CAP protein. Why is that a big deal? What does it do next? Yep, so now this binds, or I'll put here CAP, binds to the promoter. And what does that allow to happen? It allows RNA polymerase to bind and transcribe. And transcribe the genes. So even if you have number one all done in the cell, so you have this inactive repressor, it's not binding to the operator. If you don't have number two, the active CAP protein, then your genes are still turned off. So both of those have to happen together. The number one with lactose lactose inactivates the repressor. That's part of negative regulation. So, so the repressor is with negative regulation, and the cap protein, the activator, activates the genes, and that's called positive regulation. And so in this case here, you have to have the active activator and the deactive or inactive repressor to turn those on. All right? And so, all right, so let's look at the second picture to see what happens when both are present. So if, looking at this down here, this, you have lactose present and you have glucose present. So now they're both present. If they're both present, which one is the cell going to use? Glucose, right? So the Lac Operon, we want it on or off? Off. We don't want to make the enzymes to digest lactose. We're going to choose glucose instead. All right, so how does the glucose being present turn these genes off? So remember, glucose has to do with cellular respiration. So breaking down ATP into ADP into phosphate. But if glucose is present, if it's present, that means that this process here, going and putting the phosphate back onto ADP to make ATP, is going to happen. So therefore, glucose present, cellular respiration can happen. And so, so that occurs. So in this case here, there's going to be no or very little cyclic AMP because we're not going to further break down ADP because there's enough glucose to regenerate ATP and so on. So that's why it says cyclic AMP is very low when you have glucose present because you're able to regenerate ATP. So therefore, here's your inactive cap. Unlike the last picture, there's no cyclic AMP. So we have no camp that can, so therefore it's not, the cyclic AMP is not going to bind here, which means that it stays in the inactive state. So this stays in the inactive state for the cap protein, which means, can it bind to the binding site? No, so this little guy here can't, so it can't bind because it never changed shape because there was no cyclic AMP. So therefore, if the activator doesn't bind, then RNA polymerase can't bind. This is because no cap bound to the promoter. And so, in this case here, then these genes are turned off. So, and therefore, they're not going to make the enzymes to digest lactose. So lactose is present. So notice here, allolactose is still binding and inactivating the Lac repressor. So it's still not binding to the operator, but that's not the only thing that needs to turn these genes on. You need to have the active cap, and so since there's glucose present, it doesn't bind there, and so therefore, even though there's lactose present, because there's glucose, it doesn't make the enzymes to digest it. All right, and so that is the second part of positive regulation there. Okay. All righty. Any questions on that? Okay, so that is bacteria and how bacteria turn genes on and off. Bacteria have operons. Operons are only in bacteria, so there's something unique to bacteria. Our cells, we're going to look at us as a eukaryotic organism. We're made out of eukaryotic cells, and we do not have operons. We have different ways to turn our genes on and off for our cells to specialize. So we are going to do start a little bit with chapter 19. And then switch gears. Okay, so the first part of regulating and turning our genes on and off is having a good solid understanding of our DNA in our cells, which we've already talked about. We've already talked about with cell replication with division with mitosis and so on. We've talked about DNA being wrapped around proteins called histones. That's what this picture here is showing you. So here's your DNA as a double helix. And then the DNA in this picture here is just, in my case, is the blue lines here, and it's wrapped around these proteins called histones. And I use those in that analogy. It's kind of like spool, a thread around a spool to keep the DNA organized. And so a new little word here, nucleosome. Nucleosome is the protein with the DNA wrapped around it, and we'll talk about, do the definition a little bit later. This is what it actually looks like. So it looks like each one of these little dots is DNA wrapped around these histone proteins. So then what happens is, remember during cell division, as we go through cell division, the DNA condenses. And so it kind of comes together. So now you have all these nucleosomes kind of condensed together. So this is right here, one nucleosome right here. So it's a bunch of them. And then this, so you have a big long chain of these guys all condensed together, all these nucleosomes, and then they fold on one another. So like you can see this little box here is this right here, so it folds on one another. And then if you took a long strand of this, then they fold on one another. So you can see here, this is this particular picture here, and so the folds fold in on each other and eventually condense into, notice here it says metaphase chromosome, so that's during cell division. And so up here is the chromatin stage, like these kind of two pictures, and as it condenses into a chromosome, this is what it happens. And so why this is important to go over is because one of the ways that we control and turn on and off our genes is allowing access of the enzymes like RNA polymerase to get to the DNA. So if the DNA is really, really tightly packed and is all in this mixed up in these histones and so on, it makes it hard for the enzymes to get there. So one of the ways that we turn on and off genes is allowing access for the enzymes to the DNA. So that'll become important as we go through this chapter. So this part here is a little bit of a review, kind of what I just went over here. So DNA within a cell, it undergoes a variety of changes as it goes through the cell cycle. So remember, in prophase, it's more diffuse, meaning more stringy. Then as the cell prepares to divide, it becomes more and more condensed until you get that stocky chromosome. And those proteins called histones are responsible for that first level of DNA packing in the chromatin where the DNA wraps around the histone proteins. In fact, the mass of histones is about equal to the mass of DNA. So there are a lot of histone proteins that our DNA is wrapped around. So why does the DNA wrap around the histones? DNA is negatively charged, and histones, the proteins, have a high proportion of positively charged amino acids and enable easy binding of the histones to the DNA. Why? Because DNA is negative charged. Histones are proteins that have a lot of positive amino acids. Opposites attract, so the positive and negative charges attract, which is why the DNA wraps around the proteins. Histones play a very important role in organizing DNA, and they're very good at it, acting as that spool. Thus, this is a likely reason why histone genes have been conserved throughout the generations in the course of evolution. The structure of histones are very similar among eukaryotes as well as eukaryotes and prokaryotes.